So, welcome to the next training session on SSbD from the Planets project. Today we are focusing again within our series of safety dimension related trainings on safety. And I shall say thanks to the people who are in charge of preparing for this entire series of events on the safety dimension within SSbD, starting with the colleagues from QSA Lab in Poland, Shimon and Anita. From BASF, Aisha, James, and Philip are helping us here, collecting content. Cecile, Josephine, Nienke, and Marie from CA, Susan and Neeraj from TNO, Ponima from the Radboud University, Beatrice is always supporting us with interactivity tools and assessments from BNN. And my team consists of Mario, Laura, Sabine, Norbert, and myself. So welcome. Let's just dive into. Today we have a very interesting next step in the safety dimension. So after a very short recap of the session one, which we did past week on the background, basically, so really starting at the A and B of safety assessment, we will dive into in silico methodology. And as we did already have a QSAR training, so you can look it up in our resources of materials, we are going to focusing a little bit more on the relevance for regulation in the context of QSARs here today. So in silico methods for hazard assessment. And then we dive into in silico models for release and exposure. We will also see the Stoffen manager and Neeraj. Then Ponima is going to dive into environmental safety modeling. There we also have a very quick look into use talks. And Philip is then showing us PBTK modeling, which is very relevant for reverse dosimetry extrapolation in vitro to in vivo, actually. Mario is then taking it further from this modeling focus to first assessments. So migration and release, how to determine. And he's going to use examples from the food contact materials and medical devices area. And then Aisha will give us a quite good look and well representing quite a number of different in chemical new approach methodologies and look into process-related safety assessment. Finally, I will wrap up again and then also a little bit explain what the way forward will be, what the next training is going to be showing us. Of course, we as always try to involve also some interactivity or enable interactivity, but today we are not going to go really into breakout sessions, but just use Menti related questions to involve the audience. So I'm looking forward now to the training session. As a short recap from last time, we really started at the beginning, getting even that far down to the medieval times, looking into the Paracelsus law, defining safety as the reverse of risk, and risk being kind of the multiplier for from hazard times exposure. We looked into some really important terminology. We saw some dose response curves. We did also a little bit on interactivity there. Saw differences between causality and correlation. Typical exposure routes, acute versus chronic exposure, for example, quite relevant difference. Then we looked into material data, safety data sheets. We had a quite extensive and nice interactive session on a mural on MSDS. Saw some first hazard banding tools. More will come, and we will see also next time after the summer break. And we dealt with H phrases. This means that are also implemented in the globally harmonized system and also are implemented within classification, label, and packaging, CLP. Susan from TNO then introduced us to SIS, Substance Information System database, and also to some grouping and read across, not of course into too much detail, as we did not have the time for that here. Finally, James from BASF gave us insight into regulatory toxicology and current practice in industry. So with this, I would like to give the screen basically to Shimon from QSA Lab, and you're now going to be diving into in silico methods and their regulatory relevance.
So one more time, good afternoon, everyone. Today I'll be walking you through our guideline for utilizing in silico tools within the SSbD framework. And in the beginning, let's start with the foundation. Here we have illustrated the JRC revised framework from 2025 for chemical and material assessment. The core philosophy is a tiered approach, and we apply this to both exposure and hazard assessments to ensure we use resources efficiently and minimize animal testing. And the hazard identification is structured as follows. For the tier 1, we focus on screening. This means leveraging what we already have by screening the safety data sheets, existing databases, user models, and read-across data. If tier 1 does not provide enough certainty, we move to tier 2, which utilizes new approach methodologies, NAMs for short, and in vitro methods for specific endpoints. Only when necessary, we proceed to tier 3, which involves more comprehensive in vivo studies or integrated approaches to testing and assessment, IATAs for short. By starting simple and progressing to complexity only when required, we make the SSbD process both practical and rigorous. So we have come up with intuitive guideline on how to use computational tools in SSbD framework. Here we show its outline as a 4-step iterative process. At first, we start with the problem formulation, which is focused on defining the chemical structures and identifying the relevant safety endpoints. Next, we move to tier 1 assessment, where we conduct preliminary hazard screening to see what data we have and where we have the data gaps. Then we move to tier 2, where we perform a detailed hazard assessment, specifically aimed at filling those data gaps that we identified in the previous step. And here we heavily rely on in silico models. And if it's needed, we move up to higher tier 3 and higher tiers to complete the assessment. And if we discover that the compound of interest is too hazardous, we move back to the beginning for designing the chemical structure. That's why we called it that iterative process. From step 1 to 2 to 3, we just can move in an iterative way. Now let's zoom into each step individually to clearly describe how it works. So the very first step is the problem formulation. At this stage, our main aim is to identify chemicals of concern, so we can redesign or replace them by designing new similar ones. And first, we provide the candidate chemical structure for our in silico assessment. This requires standardized identifiers. Most commonly would be SMILES notation or CAS number. Next, we define the relevant endpoints. Hazard assessment must follow the European Union SSbD recommendation. This means utilizing the hierarchical REACH and CLP classifications to cover human health, environmental impacts, and physical properties. So the outcome of this stage is the problem which is the problem formulation, is a defined set of structures and established evaluation endpoints for both tier 1, which will be our initial screening, and tier 2, which will be our detailed assessment, all based on regulations and chemical intended application. Once the problem is formulated, we enter tier 1. This is the preliminary screening phase, and as you can see, it's in our guideline, it's split into 2 pathways. First one, the upper one, is about identifying data gaps. We assess online databases and scientific literature and search for existing experimental data for the endpoints of interest. And crucially, we need to evaluate the quality of the data that we export from the online available sources. The second pathway is about identifying structural properties of concern. If we lack experimental data, we use basic in silico screening tools to look for structural alerts. We identify potential hazard warnings based purely on the chemical's shape and composition, and then evaluate those results. So the outcome of this step is a clear map of our data landscape. We have completed our preliminary screening and precisely defined of our safety relevant data gaps. Once the data gaps are identified from the tier 1, they become our focus for the next tier. Thus, aim of this step is to fill them as effectively as possible. And to make it possible and fill all the missing endpoints values, our first line of defense are the advanced in silico QSR-based tools. As you can see on the upper pathway, which is the main pathway in our guideline, it's it requires a rigorous process. At first, we need to carefully choose an appropriate QSR tool. By this, we mean the tool that can cover the endpoints of interest. Then we must check the tool's documentation and verify its validity. By this, we ensure our chemical falls within the applicability domain of the tool. Which is also very important. And the applicability domain is a chemical space that was created by the chemicals that were used for training the model. And only then we run the predictions and, of course, evaluate them. If QSR models cannot reliably fill the gaps, we move to the bottom pathway. In our graph, it's not bottom pathway, which is targeted for in vitro or in vivo experimental assays. Generally speaking, the outcome of the tier 2 of our guideline is fully defined hazard profile with filled data gaps. If the substance proves to be very hazardous at this stage, we trigger the iteration process, as I said before, and go back to the beginning to consider safer alternatives. And finally, it is very crucial to understand that using in silico tools in SSbD framework is not just an exercise, it has legal and regulatory implications. So if we want regulators to accept our QSR predictions, we must follow established guidelines. In 2004, the OECD agreed on 5 core principles for the validation of QSR models. For a model to be accepted, it must have a defined endpoint, an unambiguous algorithm, defined domain of applicability, appropriate measures of appropriate statistics, and if it is possible, a mechanistic interpretation. This framework is evolving constantly, and as highlighted on the bottom right, in 2023, the OECD has provided updated guidance and practical checklist for assessing multiple QSR predictions. Adhering to these principles, this ensures that our SSbD process is robust, transparent, and regulatorily ready. So by this, we can move on to the Menti section, and I will hand out the screen to Beatrice.
The first question is what is the main purpose of using QSR-based computational models in early tier 1 safety screening? And you have different options to physically test chemical samples using lab robots, to predict a chemical's toxicity using math and computer algorithms based purely on its molecular structure, to analyze historical patient health records to find disease trends, or to simulate how chemicals physically break down in outer space.
There are no other votes than this one, so I think there's no need for comment. We can move on.
You were all correct. Okay, then we continue with the next one. Why the SSbD practitioner must check a QSR model's applicability domain before trusting its prediction. Because the model's validity depends on whether the target substance is sufficiently similar to the chemicals used to develop the model. To ensure the chemical can be legally manufactured in a specific country, chemical leaks during industrial transport, to calculate the computing cost before running the software and to check if the computer hardware is powerful enough to run the simulation. I continue with the next question. Why do toxicologists use computer tools to scan a newly designed chemical for structural alerts, to find weak points in physical storage tanks that could cause leaks, to automatically alert project managers about raw material shortages, to quickly spot known molecular fragments that have a proven history of causing toxicity, or to simulate how the chemical digests in the human stomach over 24 hours? I think I can already show the correct answer.
So if people watching the recording here, please go to our training resources. You will find QSAR training material. Yeah, and with this, now we dive into the methods for release and exposure, and I give the word to Neeraj Shandilia from TNO.
All right, thank you. So this part of the presentation is about the in silico methods for release and exposure assessment. Now, over the next 10 to 15 minutes, we will be covering a range of the topics. So we would try to understand or recognize why exposure assessment matters. What is release? What is exposure? So very basic understanding of the concept. Identify what could be the common exposure pathways. So how can you get exposed? What does the term in silico exposure assessment really means? How do we use these models to estimate release and exposure? And finally, see what sort of strengths and limitations are we dealing with when we talk about these computational approaches in silico methods. So first of all, why exposure assessment matters. Now, I think we dealt with it quite clearly in the session one also, that hazard alone does not determine risk. So risk depends on both exposure and hazard. And that's why exposure assessment matters. Now, when you do the exposure assessment, you basically answer a series of questions. The first question is, who is exposed? To what extent someone is exposed? How much? How often? And through which route? And everyday examples that you could include for the exposure could be like when you're using a cleaning product, so you're cleaning your table, your kitchen counter, something like that, the cosmetics that you're applying to your body, the industrial chemicals that someone is using, and even in the food packaging industry. So these are very common examples in our day-to-day lives that we can find the exposure examples. Now, very basic way of approaching this entire subject of the exposure is that, first of all, we talk about the source. Where does the chemical come from? So this could be the chemical used in household cleaner products. Then there comes the release. So basically, how much is being emitted from the product to the environment? So it could be emission to the air, it could be emission to the water or to the soil. And then once it is emitted out, it's all about its fate. So what's basically happening to the chemical in the environment? Is it diluting further, or is it sedimenting or depositing on a certain surface? Something like that. So this is all about the environmental fate. Then we come to the exposure. So once the entire transportation, dilution, whatsoever is done, then we get exposed to that. Now, this exposure could be through air, through water, or through the food that we are consuming, etc., etc. And finally, we talk about the potential effects, so the risk part. Now, the exposure pathways, there could be many different sources of the exposure routes. So there could be inhalation exposure. So you're breathing in the air in which a contaminant or chemical is present. You are depositing your on your skin, for example. So the direct deposition can be through cosmetic products like sunscreen or deodorants, etc. Or it could be something like you are spraying in the your spray painting, for example, and the droplets, they unintentionally drop on your skin. So there might be dermal exposure over there. Oral ingestion could also be your intentional or unintentional. So intentional can be your medicine that you're consuming. However, you could also be eating a food which has been contaminated through the food packaging. Mario will talk about it later, but this could be also one of the exposure route there. We need to also consider different populations because all people, they are not exposed in the same manner. So a worker could be exposed in a different manner than the consumer. So worker is olfactory floor settings. Consumers, they are sitting in their households. General public, they are mainly exposed through indirect ways. So for example, consuming the water, which has been somehow contaminated priorly from your local water reservoir, for example. Environmental species, so environmental exposure is also quite important, and Purnima will talk about it also later. And once you have, depending on the chemical to chemical, there are a certain degree of permeability of the chemical inside the body. They could deposit into the lungs, skin, or mouth, depending on the exposure route. Now, to start with the exposure assessment, the first and most important thing is to establish these exposure scenarios. What if this happens, then this will happen, something like that. So any exposure scenario, any exposure depends on the phase chem properties. So the parameters related to the chemical or the substance itself, as well as its process or use conditions. So for example, it could be the industrial processing or use, consumer product use, or accidental release. So we need to specify exactly in what sort of process conditions or use conditions the substance is really in. And these use conditions can be, for example, how much amount of the substance that we're using, or the concentration of the substance, what is the frequency of the use, or the duration of the use, sort of ventilation in our houses or at the factory hall, are we having some sort of protective equipment or not? Those type of, all these elements we take into consideration while defining the exposure scenario.
Here we go with our Menti question. Imagine you walk into two different workplaces for 10 minutes. Room A could be someone is actively spraying a bathroom cleaner in a small, poorly ventilated room, and room B, a chemical is being used inside a fully closed industrial system operated automatically. And now the question related. Sorry, my mouse doesn't work. In which of both situations, and here now you can see them, could likely lead to higher inhalation exposure for a person in the room. In room A, the one which is actively sprayed with a bathroom cleaner, and it is a small poorly ventilated room, or room B, where a chemical is being used inside a fully closed industrial system operated automatically. You have 4 possible answers: room A, room B, both about the same, or impossible to tell.
So it's one person, 2 people for room B and 1 person for, okay. So let's go to the next question and see, okay.
Okay.
There we can really talk about, okay.
This is an open-ended question, so you can write what factors influenced your choice in the previous question, why you decided it is room A, room B, or impossible to decide.
Okay, so largely people do go for room A. Room B, I think there were 2 votes which were there, and I believe that has to do something with this question, are they volatile or something? Small room, higher dose. That's nice. Okay, well, now since we have established that we have a common understanding of what is exposure, how this thing generally works, I will go through the workflow. How do we assess the exposure? So first, it all starts with defining the chemical. By chemical, it could be any substance. So I just included chemical here, but it could be anything. And what are these use conditions? So what are sort of properties? What is the use pattern and how much amount is used? We then estimate the release. the release to the air, to the water, or to the land. Now, if it's a volatile, then it could be fumes coming out of it, or aerosol coming out of it. If it's a dusty, then powder particles getting airborne, for example. Water discharge could be that you are directly discharging into the wastewaters, or some accidental spills are happening in the groundwater or something like that. In the land also, it could be the waste, or you are landfilling it. So these are the type of the release that you can estimate at the second step. Third step is all about environmental fate. Now it is in contact with the environment. What's going to happen with it? So there are different environmental fate models. So for example, use talks, which Purnima will talk about. For indoor air modeling, we have different types of the model, which talks about the dilution happening, sedimentation happening, what's happening in the air. Same goes for the soil. Here we talk about indoor air, outdoor air, etc., etc. Then based on that, the environmental fate, the exposure concentrations are determined in air, in water, and in soil. So this is basically the exposure concentration to which a certain person or an environmental species is prone to inhale or to absorb in their skin or into their body. And finally, the characterization, risk characterization. So what we do is, once we have this exposure concentration, we compare it with established regulatory limits. Now, this has been talked about in the session 1. For different chemicals, we have regulatory established limits. They could be occupational exposure limits, or they could be some other reference values determined by the European government, or European Commission, sorry, or it could be some national governments or something like that. And if the exposure concentration is lower than these regulatory limits, then it's an acceptable risk. But if not, then it becomes an unacceptable risk, meaning that we need to make some changes in the product or in the process to lower down the exposure. So it's sort of an iterative process where we keep on refining the product properties and the process properties. In silico exposure. So in silico, I think it's quite clear, is based mainly the computer-based prediction or simulation. Examples, they include mathematical or computational models. They could be even database, some machine learning, QSAR tools, etc. In silico methods, these computational tools, they're quite fast, lower cost, and they could be used during early screening. So that's why they are quite advantageous in the entire scenario of the safety and sustainability by design, especially in the early stages. And the type of the inputs that we need are chemical identities. So for example, the CAS number of a chemical or substance. It could be the physical chemical properties like vapor pressure, water solubility, log KOW, use amounts, use pattern, and population or activity information. And like any other model, in silico exposure models, they also, if you have better input, you will have better prediction. There's nothing different in that part. Now we use a different series of exposure models like Stoffen Manager, EasyTalk, FonsExpo, etc. For example, Stoffen Manager, one of the exposure assessment tool, it is used to estimate worker inhalation exposure during product manufacturing. On the other hand, on the right side of the screen, you see FonsExpo, which is used to determine consumer exposure during product use. So depending on the exposure scenario, the population and whatsoever, the applicability domains are defined, and based on that, these tools are used. These tools have different applicability domains, and it all depends on the exposure scenario that you are dealing with. And for your information, within Planet's task 2.1, there is a deliverable 2.1 in which we have established this comprehensive state-of-the-art inventory of the methods and tools, where we have divided these tools in different scenarios that one can encounter as part of the case studies, as well as outside of the case studies of Planets. One of the tools, Stoffen Manager, very widely used. I wanted to give you a very quick insight into it. So this is the interface, that how you look. So this, you start with a dashboard here. You create a product, meaning you define the PhysChem properties, and you create a scenario, meaning you define the process or the use conditions itself. The governing part is this equation. This is a very long equation, but I will ease in it for you. CT is the concentration, the exposure concentration. It depends on the intrinsic emission score. So basically the volatility of the substance or the dustiness of the substance. So basically how much can it emit, the substance itself. It depends on the activity. So the factor H, handling task score, meaning how much energetic process are you using? If there are local control multipliers present or not. So for example, if there is a local exhaust ventilation or you're making a complete barrier or containing your source. If there are general ventilation multipliers, so you, for example, on the factory floors, you have these central exhaust ducts that you have. So these are the general ventilation. Then there could be the impact of the background. So for example, something is happening in the far away, in the far field, like on the other side of your factory floor, where you have some sort of cross-contamination happening in your own activity. Or there could be some contamination or unclean workforces. So example, imagine some accidental spills happened on your factory floor and they dried over the time and a forklift goes over it or a worker goes over it. And this dried spill becomes, the particles start coming out of it and they become resuspended into the air. And this can be again inhaled by the worker. So this also impacts your exposure concentration, net exposure concentration. So Stoffer Manager takes into account all these factors and provide you at the end this exposure concentration. Finally, strengths and limitation. I don't want to spend much time on it because this is not unique to them. They're quite fast, very cost effective. The good part is that they support early decision making, so we don't have to delve into the high cost sensors, lengthy process of measurements. We can use these computational methodologies to quickly, with certain uncertainty, determine the exposure concentrations and make our decisions right away. However, on the other side, the biggest limitation, I would say, for these exposure assessment models are the assumptions, and also sometimes some of the assumptions, these are quite far from the real life behavior. So for example, the worker breathing rate, we have a set fixed of breathing rate of a worker. So depending on how the work is being carried out, this breathing rate might change from person to person, and also how the work shift might change. So there are certain assumptions that these models make that we have to be aware of that while making the decision, and make the decisions accordingly based on the output that we get. And of course, the uncertainty. That's it from my side. Thank you.
Thanks a lot, Neeraj. So let's flip over right to Purnima.
Yeah.
More on exposure modeling. Thanks, Purnima.
Today I will speak about environmental exposure, or like largely also on exposure modeling. Also looking at Neeraj's talk, he already covered a large part of it. We already know that chemicals after use, they can be emitted into the environment. They can go through different fate and transport process. Based on that, they have different environmental concentration depending on where they are released. If they end up in air, water, soil, or sediments. Why is it actually important once these chemicals are emitted into the environment? These environmental concentration can cause exposure. It could be for the ecosystem or for the humans. So it makes it extremely important to know what is the exposure. But to do these actual measurements, because it can vary in space and in time, so it's hard to actually make measurements or tests. So largely we use models. There are several models that you already saw from the presentation from Neeraj. It could also vary depending on the purpose, which model is used. So I will briefly explain why are models used, and I will also give an example of Fuse-Tox model. Why do we use exposure models? Actually, first to know where do chemicals go once they are emitted, and how long do they remain in the environment. And based on this, we can also see like who or what is exposed to actually look at the risk. And the exposure depends on several factors. The key ones are the actual chemical properties, how persistent a chemical is, how mobile that can actually tell if it would end up in the surface water, or if it's more volatile, if it's mostly in the air, and also on the environmental conditions, if it's emitted to air, water, or soil. And they can also, depending on these physicochemical properties, we can also determine there is frequent transport between the different environmental compartments, so we can actually assess where would the chemical actually end up and the actual concentrations. So to do so, there are several models. Basically, most models actually do the first part. They look at the chemical fate to estimate the concentrations. And then based on these concentration, actually, they assess the impact or the exposure. So the example that I would talk about today is USTOX. USTOX is intended as a life cycle impact assessment model. So they do, it has several components. So it has a fate modeling component, an exposure part, and also based on that, it also assesses the effect that causes on the environment or human. When I speak about human exposure, I would like to also make it clear the exposure can be both direct exposure, like if you inhale, or it could be like indirect exposure that some of the species are exposed to, like fishes are exposed to certain chemicals. It's bioavailable in their system, and when we eat those fishes, then we are actually also exposed to these chemicals. So I will speak about both direct and indirect human exposure. So the first part is the fate modeling. This is the what you see is a USTOX nested component structure. Because it's hard to do it spatially explicit, they look at the different environmental components and also looking at indoor, urban air, and on continental and global scale. What do we see? What does it actually estimate? It looks at the emission compartment, if the chemical is in air, water, soil, or substance after it's released. And it also accounts for the residence time, how much after release will a substance actually remain in the air, or how much residence time will it stay in soil. And it also look at, like, these arrows kind of also indicate the transport process, which can mean that the substance can move between the environmental compartments. Two main transport processes are usually considered. The first one is advective transport. It's the movement between different compartments, for example, between, let's say, between air and water, like certain substances can be deposited in the agricultural soil. From there, when it rains, it can be transported with the runoff to fresh water. And if there are certain substances, they can also volatilize and go back again to air. So there can be advective transport through movement, or diffusive transport, because there is a concentration gradient between in this concentrations between the different environmental concentrations. So the substance can also move between different environmental compartments. And it also accounts for not just that substances are in the environment, it can also be removed from the environmental system through different removal process. The few main ones are like through degradation or biodegradation by microorganisms. It can bury in the sediment, or it can leach through several layers of the soil and reach the groundwater, or it can also transfer back to the stratosphere through these transport processes. So this is the conceptual part of the fate modeling, how we, in the model, how do we get from actual emissions to concentrations. And the next part, like I said, there is the environmental fate part, and the next is to look at the exposure and the effect. So the first example that I will speak about is the environmental exposure modeling. As you already see, the amount of chemical that's emitted based on the fate modeling framework that I showed, you can actually estimate environmental concentration in air, aquatic, and also terrestrial ecosystem. That's called the fate factor. And once there are environmental concentrations, they can also become bioavailable. For example, if there are concentrations of a certain chemical in fresh water, it is also readily available for aquatic species, and they can take up. So that there we can calculate the ecosystem exposure factor. once the species are exposed, they can be affected based on the ecotoxicity of the substance. So we can calculate the potentially affected fraction of the species, which can be done on like individual species level and on different trophic levels, so that it actually accounts for the whole ecosystem. And based on that, we can derive the ecotoxicity effect factor, like percentage of affected fraction in cubic meter per kg of substance emitted. And the last part is kind of like a severity factor that kind of shows, like based on these toxicity effect, how severely the ecosystem quality is damaged. Based on this, we can calculate the ecosystem quality severity factor. And like I said, like when there is emission, it can be the exposure can be for humans or the ecosystem. So the next part, we look at the human exposure. Like I said, like once there is environmental concentrations of these chemicals, it can be either in air, water, or soil. We have the fate factor. Based on that, humans are also exposed to these environmental concentrations, which could be through, you also saw the different exposure routes, which could be through inhalation, that you inhale certain concentration of certain substance, or you consume a certain substance through water or food ingestion. Based on that, we can derive the human exposure factor similar to the ecosystem exposure factor. But in this, after this, for the case of humans, we can also look at diseases incidence in exposed population. That means like based on the dose response factor, how much of the population is at risk for cancer or non-cancer related diseases. And based on that, we can also estimate the human health severity factor, that actually means toxicity related damage to actual human health. So it provides an important significant input towards understanding like how much a substance emitted can affect the human exposure. This is actually the functioning or framework of the use tox model, but I will next give an insight of how the model actually works. This is how I have used the newest version of the use tox model. I think the 3.0 version, and it's a Excel based user interface, very easy to use, and it's also publicly available and free to use. I've also linked it to the use tox model. It's also they have a lot of guidelines on the documentation, user manual, how to use it, and also elaborate tutorials on use of use tox model if you're interested. And like I in the beginning mentioned, like the purpose of use tox model is for life cycle impact assessment. So it is compatible with current life cycle assessment workflows, and it is also very much used for the SSbD. And if you look at, if you go from the first interface, this is how we would actually calculate the fate and also the exposure and effect. This example here is for the case of building materials. As you can see, you have to add the substance information if you're looking to estimate the effect for a certain substance. You can they have a database with 10,000 chemicals that are listed. It's also possible that the chemical you're trying to look at is already on the database. And the most important part is also to add the emission information. This is a important input that goes into the use tox model. And you can also classify what is the product type, or specify the consumer product scenario in a way. For example, if we speak about building material, like the exposure to ecosystem will only happen once they are emitted into once they are in the landfill and where they can leach into surface waters. But whereas in contact materials, like the human exposure can actually happen through migration, which you will also hear later in the later part of the workshop. So you can also define the specific exposure scenario, and based on that, the model can actually already estimate the mass that would end up in the environmental compartments, and the different fate factors, and the effect, the potentially affected fraction of the species. And similarly, in the same interface, you can also calculate human exposure. It's the same component. There is substance information and the emission information that needs to be inputted. Based on that, you can actually calculate the human intake fraction, which is basically that you can assess how much you can the total amount that can be inhaled or through ingestion or also through dermal exposure. And here you can also see if it's on the continental scale or global scale. Yeah, you can also calculate the effect factor and the severity factor based on the conceptual framework. So as a brief example use talks with this model, you can also estimate both human and ecosystem exposure and also what kind of impact it causes. That's it from my side.
Thank you, Purnima. And of course, we need to translate exposure to doses inside the body. And this is now presented by Philipp Demuth from BASF. Philipp, the stage is yours.
Yeah. As already introduced, we are now interested into how we can relate external exposure to internal exposure. And doing that without the use of animals can be done by using physiology-based toxicokinetic models. For this reason, I would like to first start with a quick introduction into toxicokinetics. So as opposed to toxicodynamics, toxicokinetics investigate how an external dose or exposure relates to an internal dose. So in general, what the organism does with the substance. This is opposed to toxicodynamics, where we are interested in the interaction with of the substance with the organism that can lead, for example, to adverse effects. So with this information, we can now decide if a potential compound that might cause hazard in an in vitro setting, for example, is also of relevance for organism level effects. So if an external exposure does also can induce, for example, cellular or subcellular changes in the organism. And to better understand this, we need to address certain aspects of the toxicokinetics. And those are in general subdivided into the so-called ADME. So this abbreviation stands for absorption, distribution, metabolism, and excretion. And I would like to quickly guide you through these different steps. So first we have absorption. And this is dependent on the administration or exposure route and can be in general subdivided into inhalation exposure, oral exposure, and dermal exposure. But it can also, for example, include intravenous exposure if we are looking at a, for example, pharmacological compound. The absorption does also depend on the preparation. For example, you can imagine that if you have a certain compound in food, for example, this might lead to different uptake kinetics than when the compound is available via drinking water. And in the end, the absorption also depends on the dose, because at a certain dose, we have, or we might have saturation mechanisms that lead to a plateau in the uptake of the substance. Then, in the next step, we investigate the distribution of the compound. And this, in general, depends on the substance lipo or hydrophilicity, but can also be affected, for example, by active mechanisms like transporters. Then an important step is the metabolism of the compound. So here we can differentiate between the qualitative aspects of this metabolism. So the question, what kind of metabolites are formed, for example, in the liver, but metabolism can also take place at other sites, so in other organs, but also the quantitative aspects of the metabolism. So is the substance metabolized at all? And what are the kinetics? So how high is the rate of metabolism? And then finally, we can take a look at the excretion of the compound. And here also qualitative aspects apply. So for example, what are the pathways that play a role? Is it via the kidneys, the liver, or maybe for a volatile substance, also the lung? And how high, again, is the rate of excretion? So it can be very slow for certain compounds or really fast for others. And if we have information about those parameters, we can then take a look or simulate or whatever, understand the concentration time curve. And the concentration time curve shows how those different parameters interplay with each other. And based on this curve, we can deviate certain parameters. So one important parameter is the Cmax, or the maximum concentration that is reached after a certain dose. So this dose could, for example, be 100 milligram per kilogram body weight. And depending on the, for example, absorption rate of the compound, we reach a certain internal dose. And normally you look at the maximum concentration in the plot plasma. And then after a single dose, for example, the excretion of the substance, but also the metabolism, then leads to a decrease of the internal concentration over time. And here, one important parameter is the half-life. So what is the time required for the concentration to decrease by 50% compared to the Cmax? And this also is highly dependent on the compound. Then again, we can have a look at the area under the curve. So this is the overall systemic exposure that takes place. For many toxic compounds or for pharmaceutical compounds, we need to pass a certain threshold to induce an effect. But for other compounds, especially for carcinogens, the area under the curve might apply as an important dose metric. So with that, I would like to give you a quick overview about the relevance of toxicokinetics within toxicology. So one important aspect is the accumulation of compounds that might occur, but also the question of species differences. So if we have certain effects in the rat, but not in the human, this can be due to toxicokinetics. We might have overload or saturation phenomena. And one important aspect, I think also within this project, is the data that is used for risk extrapolation to humans, especially when it comes to in vitro or in silico results. And the big question is, to which degree do we have to rely on in vivo studies to assess these parameters? And here's where PBDK models come into play. So PBDK models calculate the plot plasma and the tissue concentrations of a compound based on physicochemical data and certain in vitro input parameters. And this is normally done as a function of time. So the input parameters can again be subdivided. On one hand, we have physiological input parameters, and those depend on the organism the PBDK model was developed for. So, for example, for a human, male human or female human, I have other physiological input parameters than for the rat. And those input parameters include the body weight, the tissue volumes, and stuff like that. And those are typically found in the literature and are implemented when such a model is developed. And also here it depends on the complexity of the model, how many compartments are incorporated. I can have a very simple one compartment model where I just assume that the organism is one huge compartment, but I can also have very complex models like this 9-compartment model, or there are up to 15 or 20 compartment models, depending on the toxicological question you want to address. So for example, if you want to model the exposure in a certain organ. Then you have physicochemical input parameters, and those depend on the chemical. So this could be molecular weight, log P, or the tissue partition coefficients, which do mostly also depend on the log P and the polarity of the compound. And then you have kinetic or metabolic input parameters, and those depend on the compound, but also on the organism. So this can, for example, be in the intrinsic clearance, which gives you information about how good the compound is metabolized. And one important thing to say about those models is that they are always only developed for one compound or substance. And if you have a metabolite that is formed during this process, you have to apply an additional model for this metabolite. So you only do this for one particular compound. And then as a readout, you get the concentrations of the compound in the different compartments. And we are especially interested in the Cmax value in the plasma. And for the different compartments, the results could look like this. So again, you get concentration time curves, and depending on the lipophilicity of the compartment and certain parameters that might also play a role, you get different concentrations and concentration time curves. So one important differentiation which I have to make here is the one between bottom-up and generic PBDK models. So PBDK models were or are used a lot in the pharma industry. And here, normally bottom-up pharmaceutical PBDK models are applied. You have a lot of information about the in vivo ADME parameters, parameters of the compound, and also the in vivo toxicokinetics. And therefore, you can really fine-tune your model dependent on the actual data. In toxicology, we normally use generic PBDK models. Here we have, as I explained, a predefined physiological structure. It is applicable to a lot of chemicals, not all, but a lot, depending on the parameters. And you normally only use in vitro or in silico data for the parameterization. And therefore, for our purpose, generic PBDK models are normally applied. So I would like to quickly go through the toxicokinetic input parameters that are needed. So on one hand, as mentioned, we have the hepatic clearance, and this can be experimentally assessed via incubating the test compound, for example, with human hepatocytes, and then analyzing the concentration and the decrease of the mother compound over time, for example, by LC-MS. And this gives you information about how good the compound is metabolized, especially in the liver. Then you have plasma protein binding as an important parameter. And this gives you information about the fraction of the compound that is free in the blood plasma, because depending on the structure and the polarity, a huge amount of the compound can be bound to proteins. Then you have the permeability as an important parameter after oral uptake. For example, in inhalation, you might also incorporate this parameter, but it's mainly important for oral and dermal uptake. And here you can apply certain in vitro systems, for example, KK2 cells, which are cultivated as a monolayer. And then you measure the uptake of the compound across this monolayer in vitro. And this gives you an input parameter for your model and information about how good the model is taken up via the gut, for example. And then finally, you have the tissue blood partition coefficients, and those give you an idea in the model itself in which compartments the compound might end up. So for example, a highly lipophilic compound might accumulate in the fat compartment, while a hydrophilic compound is highly present in the blood plasma. So now I would like to quickly give you an insight into one model that is accessible via the public domain. I hope you can all see my browser now. And what you see is the so-called integrated chemical environment, which is provided by the National Toxicology Program of the US. And here we have a open source user interface to use the model HTTK, which was developed by the US EPA, and which is a quite simple generic model that can be used for high throughput purposes. And here you have the possibility to put in certain parameters for your model simulation. And first, you can, but you do not have to, depending on the substance, provide the aforementioned physicochemical and toxicokinetic parameters. If you have compounds where the data is accessible via the database that is part of the interface, you are fine. In other cases, you have to upload it here. So I would like to get into two compounds, diazepam and acetyl salicylic acid, two drugs, two painkillers, which have highly different toxicokinetics. So luckily for those compounds, we have the input parameters already in the database. So we just have to provide the model parameters. So we have human as a species. We assume a body weight of 70 kilogram and an exposure dose of 1 milligram per kilogram body weight. We have an oral dose and an exposure interval of 24 hours, which we are looking at. We first simulate the time course of 1 day, and as an output, we get the concentration in micromolar. So if we now run our model, we get the already mentioned plasma concentration time curves. And here we have for both compounds different curves, depending on the compartment that is applied within the model. We can now choose the concentration in the venous blood as an output, and we nicely see two highly different concentration time curves. Aspirin is, or acetyl salicylic acid, is cleared already nearly completely after 15 hours or 10 hours, while diazepam shows a high accumulation and nearly no decrease after 1 day. So if we now, as an input, apply a typical duration of a toxicological chronic study of 90 days, in the rat, for example, we would then see the corresponding plasma time curves that give you information about how the compound would behave after chronic exposure. And again, we choose the venous concentration as an input. And what we see is that for aspirin, we have nearly no change in the concentration after 1 milligram per kilogram body weight, even if we are exposed chronically, while for diazepam, we have a 15 or 10-fold higher internal dose due to the accumulation of the compound. So this gives us a nice insight into how toxicokinetics affect how the external exposure relates to an internal exposure. So what can we use this data for? And this is normally, for us in vitro toxicologists, the so-called in vitro to in vivo extrapolation, or IVIV. So like I just showed, we would first define a starting dose. This can be an arbitrary dose, for example, of 200 milligram per kilogram body weight. We then would calculate the target concentration with our PBDK model. And this is normally the Cmax concentration in the blood plasma, because this is what we relate to the in vitro concentration that we test in our assay. And this could be, for this particular example, 400 micromolar. We then would take our in vitro effect concentration, so what we measure in our, for example, cytotoxicity assay, and this example is 50 micromolar. We can then use the rule of 3 to calculate to which external exposure this in vitro test concentration would relate. In our case, in our example, this is 25 milligram per kilogram body weight. So this would be our calculated in vivo point of departure. And this is something we can now compare with an external dose, for example, at the workplace, which in this case would be 30 milligram per kilogram body weight. So we would see that our in vitro point of departure does not surpass the external dose, or other way around, sorry. The external dose is higher than the internal point of departure, and therefore we would assume that there could be some kind of problem. So with this, I'm at the end of my presentation, and I will quickly quit the sharing.
Thank you very much, Philip. This is very insightful. And now we need to see how do we get the doses and can we measure it when we also look at final products. And therefore, I give the word now over to Mario, who will drag us into how to determine migration and release.
Yeah, thanks a lot and welcome. Yeah, as Martin said, we already heard a lot about exposure in silico models now, and now we're more interested in the substances and materials that will end up at the end in our bodies, bloodstream, digestive tract, and other organs. So since plastic is highly used for food packaging and medical devices, migration from those materials is increasing a current problem. And the focus will mostly be on those two examples today for the next few minutes. So around 60% of the global produced plastic is ending up in landfills, and about 1% is ending up in the sea. So pictures like this on the right side, they will become more and more common since this plastic production is expected to double until 2050. Logically, also the exposure to those substances and materials derived from those packaging material and medical devices is highly increasing in the upcoming years. So since plastic products or plastic is not a single material, but rather a chemical complex mixture containing thousands of substances like plasticizers, flame retardants, or UV stabilizers to give the end product its stability and function. Those substances can be released during its lifetime, mostly due to two different scenarios, the environmental degradation and release and the use phase degradation and release. So during the environmental degradation and release, UV radiation or the humidity and water exposure or oxygen, ozone, mechanical stress and temperature extremes can lead to the migration and forming of microplastic or nanoplastic or the release of potential substances of concern. Then during the use phase degradation and release, so the repeated heating, for example, or cooling, freezing, the microwaving of the products. So if we put like from the delivery service, the plastic stuff into the microwave, it can be it's highly likely that materials are leaching out of substances are leaching out of those materials. Or also if we put the plastic products into the cleaning into the dishwasher, it might be that the plastic products are aging and then microplastic or nanoplastic are released. What is also a very important point is the contact to acidic, fatty, salty, alcoholic or hot foods, which I will mention later, because this can also leach out important or substantive concern. And of course, the mechanical wear. So during the use, then the scratching, flexing, or if we opening or closing a plastic box for several times, this can also release unwanted micro nanoplastics. So here I want to show you an example of migration from food contact materials. So in this case, baby toys, as we see here. And we know our little ones like to take the toys into their mouths where the materials get into contact with the saliva. And what the researchers wanted to find out if I add artificial saliva to those samples, if some materials or if some substances are leaching out. So they incubated it 2 times for 30 minutes and it was spinning and then analyzed afterwards. So there is a similar study, did nearly the same with another simulant. So it was 50% ethanol, which was according to the European test regulations. So it was for 10 days and 60 degrees. Afterwards, they wanted to find out, okay, what was migrating from those products, and there are different approaches how to analyze this. So for example, the HPLC, high performance liquid chromatography, the FTR or GCMS, and also ICP-MS. So ICP-MS, for example, is used to measure heavy metal impurities in the product, which might end up into the human body. So another example is about plastic being used in the about plastic being used in the medical devices, so in the healthcare sector. So this sector accounts for around 2% of the global plastic production, and both studies provided here show the release of micro and nanoplastics. So for example, here we have absorbable suture, and this was soaked for 8 weeks in artificial body fluid. And we can nicely see here an electron microscopy picture where we can see released microplastic particles and nanoplastic. Then another study was about tracheostomy tubes, where they cleaned it excessively, and then they also saw that a lot of microplastic is released, and this is at the end ending up into our bodies. And then there was another study, for example, for medical devices, where they checked for leachates. So, for example, we are exposed when we are in hospital to medical cream liquid products, or to infusion bags like blood bags, or to syringes, catheters, tubes, different tubes, and also gloves. And they also wanted to find out, okay, which materials are leaching out from those products. And here you can nicely see it was categorized into 4 different groups. And what is super prominent here is DHP, which is phthalate, and it is already registered as a substance of very high concern because it's potential cancerogenic and also leading to endocrine disruption. So it has endocrine disruptive potential, but it is still used for medical devices, even if it's banned for other product categories. So the problem is it was promised, or the first regulation said, okay, this is the sunset date will be May 2025. Okay, and then they said we have to prolong this, and now the sunset date is July 2030. So until then, we are still exposed to substances of very high concern, and yeah, we have to really change it as quick as possible to find alternatives which are less problematic. So how are those migrating chemicals from fruit contact materials are be tested? So first, we need to prepare the sample. So let's say we have a thin plastic foil, and we have to cut it in a certain size according to a surface area, and then we add a certain amount of food simulants. So there are 6 different categories of food simulants shown here. So for example, ethanol, acetic acid, higher concentration of ethanol, depending on the food that you want to mimic. And then you add a certain amount based on your surface to your sample, then incubate it for a certain time. So there are like controlled test conditions available based on the European regulation. And for example, let's say we test for food contact conditions. So we tested for 4 hours at very harsh conditions for 100 degrees Celsius to mimic food contact. There is also other options, like you can also do it for 4 hours at 140 degrees for 10 days. And according to this, you have your 2 different readouts. So you have your overall migration, which you want to where you want to measure the weight from the material after the exposure is measured. So if the material lost, for example, weight because some substances were migrating into the food simulant. And then there's the specific migration where you want to know, okay, which substances are really now into my leachates. And as I said here, depending on the food, so food simulant A would be ethanol, for example, which would be for hydrophilic foods, such as many fruits, vegetable, meat, egg products, and B, for example, acetic acid. So if you have, for example, fruit juice in a plastic bottle, you want to mimic this, and then you would go for that food simulant and test with those test conditions. So, and if you have, if you are under a certain limit for the migration, then this is certified as safe for food product use or for food packaging. But there's still a lot of challenges. For example, the changes in the particle physical chemical properties, so like the size, the morphology, or the mobility, uptake per organisms, and toxicity, bioavailability, composition, which can always change during the degradation. And yeah, the biggest question then is which degradation and use case scenarios lead to which changes in release and migration behavior of which types of material. So Aisha will tell you more about now how we can test the activity of those leachates, for example, or pure chemicals. And yeah, thanks. This was my part.
Thank you very much, Mario. Good, then in chemical assessment, Aisha, stage is yours.
Thank you, Mario and Martin.
Based on published analysis of rich systematic toxicity testing, for example, reproductive, developmental, repeated dose toxicity, what is the realistic order of magnitude for animal use per endpoint category across substances? Between 2,000 and 10,000 animals is option A. Between 50,000 and 200,000 animals, between 500 and 5 million animals, or between 10 and 30 million animals.
To like when we combine the reproductive toxicity test, developmental toxicity test, and repeated dose toxicity, it is equal to 4.2 million animals required for the final proposal of the authorization. So it is 4.2 million in total.
We continue to the next question, right?
Yes, please.
In the context of alternatives to animal testing, what does the abbreviation 3Rs stand for? Replacement, reduction, refinement, or recycle, reuse, recover, reduce, reuse, replace, or rekill, resurrect, and reborn?
And this was replacement, reduction, and refinement, 3R principle. Then let me start with what in-chemical NAMs are. And according to JRC's definition, in-chemical NAMs are like abiotic methods that measure chemical reactivity. And this definition is from 2011. And these methods are used to assess the chemical safety without the need for animal testing. Rather than studying the whole organisms, they look at the intrinsic chemical properties of a substance and especially its reactivity. And these assessments, these assays measure how chemicals interact at the molecular level. For example, this can be with biomolecules such as peptides or proteins, or you can also use substances to mimic the environmental conditions that will mimic your potential exposure. And overall, they help us to identify, rank, or prioritize the potential hazards and support both the risk assessment and also as well as the product development. And through this presentation, I will introduce you some in-chemical assessment methodologies. And these methods do not represent what we specifically apply at our company, but methods from OECD guidelines or from publications. I will start with this skin sensitization guidance. A skin sensitizer is defined as a substance that induces an allergic reaction after a repeated skin contact under UNGSS classification. And this process is well described by an adverse outcome pathway, which links the initial molecular interaction to the final adverse effect. And the final step, the first step in this pathway, is the covalent bonding of chemicals to skin proteins. And this is followed by then a series of biological responses, including the activation of skin cells and the immune system. And in this OECD test no 442C, they specifically address the first key event, which is the molecular initiating step. And examples include, there are 3 tests, direct peptide reactivity assay, amino acid derivative reactivity assay, and kinetic direct peptide reactivity assay. And in this first direct peptide reactivity assay, this is a method that targets the first key event of the skin sensitization, namely this protein binding. And there, the underlying principle is that many skin sensitizers are electrophilic and can form covalent bonds with the nucleophilic sites in the skin proteins. And in this assay, you incubate the test substance with the control temperature with the synthetic peptides. This can be cysteine-containing peptide, this can be lysine-containing peptide. And after incubation, from the remaining peptide concentration measured by HPLC and UV, you are quantifying the depletion. Like if the peptide depletion is high, this means this indicates a strong reactivity and therefore a higher likelihood of skin sensitization. And then next, I will introduce reactive oxygen species assay for photoreactivity, and which is described by this OECD test no 495. And there we are checking the toxic response if it's triggered when the photoreactive chemicals are exposed to light. And it specifically distinguishes between 2 different types of reaction. First one is producing free radicals. The second one is producing the singlet oxygen. And we are doing it by exposing it to a simulated sunlight via a solar simulator. And the generation of these chemicals, the reactive species, is measured by a UV spectrometer. However, here it is important to note that this method is also limited to some chemicals. That means certain criteria, the solubility criteria should be met, and also it shouldn't interfere with this detection reactions. And in the next round, I will introduce this abiotic transformation, which is exposure relevant NAM. And this is OECD test guidance number 111. There we focus on the abiotic hydrolytic transformation of chemicals in aquatic system. This can be related to substances, the relevant to substances that can potentially enter to surface waters via use, runoff, or even atmospheric depletion. And these are conducted in aqueous buffers at different pHs, pH 7, 4, and 9. And here we are incubating the samples in the dark under controlled temperature because we don't want the photodegradation to be considered as a hydrolysis procedure here. So we are separating them. And here we measure the hydrolysis rate as a function of pH, or we identify if there are formation or decline of hydrolysis products. And we can also use here the labeled substances to track the pathway buffer, such as carbon-14. And this is also designed as a tiered testing approach. But this test is applicable to slightly volatile or water-soluble chemicals with suitable analytical methods. And the next one that I will introduce, I have previously introduced actually. This was a ferric reducing ability of serum assay, the FRAS assay. It is a simple abiotic method that is used to quantify or estimate the oxidative damage potential of the tested material. This is mostly applicable to nanomaterials, by the way. There we are measuring the total antioxidant capacity of the human blood serum and then the reduction in that antioxidant level. And in this experimental setup. We are incubating the test materials in human blood serum, again under controlled conditions. And during this process, the antioxidants present in the serum can be consumed depending on the oxidative damage potential of the chemical that we are testing. And after the incubation, we are treating the system with a ferric reduction reaction. And in this step, with the color change from the ferric ion, which is yellow here, and to the ferrous ion, which is blue, with the color change by tracking it via UV visible spectrometer, we are quantifying the oxidative damage to compare between the nanomaterials. And then the next one is the dynamic dissolution in lung sweat simulant conditions. And here we are assessing for the biodurability, dissolution, and transformation of nanomaterials or materials under physiologically relevant and dynamic conditions. In these systems, materials are continuously exposed to a flowing media, which means this better reflects the real biological conditions because we are continuously clearing the system and it's not a static test. And the key parameters that can be defined here is the half-life of the material, the release, the leachable ion content, or the dissolution rate of these materials. And hence, we are seeing if these materials are cleared from the body fluids or not. And overall, this test can bridge the gap between intrinsic material properties and the realistic scenarios, making it highly relevant for the safety assessment. And another method that I will continue with is the reactive oxygen species generation with EPR spectroscopy. There, we are generating a radical with by using a spin probe. Normally, this oxygen species are not visible, but then the spin probe traps it, and we can visualize, we can see the signal. From there, we can understand if there is an interaction between the surrounding of the chemical, and this is highly related to surface reactivity and the surface area of the chemical. Hemolysis assay is more or less similar to the FRAS assay. There we are using a sheep blood, and again we are incubating it with this nanomaterial or material dispersion. And after the incubation, if the material ruptures the cell membrane, there is a release to hemoglobin, and obviously the color of the media is changing. Then we can detect again from this color change we can estimate the hemolysis rate, the membrane disruption potential. And then I will briefly talk about the advantages and limitations of the in chemical approaches. First of all, I can say that they are relatively easy to run with controlled conditions, which leads them to be highly reproducible. And these reactivities are like chemistry driven by using organic chemistry principles. Therefore, there is a clear mechanistic understanding. And these methods are usually performed by using LCMS, HPLC, sometimes ICP, sometimes UV visible. And that's why it is highly standardized and they're practical. When we come to the limitations, these are obviously we are using here like in an artificial environment. So that's why we may not be able to mimic the protein environments. And these are also non-physiological conditions. which may need to this like the experimental setting is not really cannot be really compared to the in vivo systems, like including maybe there can be some bias. And they have a supportive role only, and they are not replacing any in vivo testing, but these are early stage tests, and it can be integrated into the testing strategies where we may have to rank or the ranking data is needed.
In the context of toxicology and alternatives to animal testing, what does the abbreviation NAM stand for? New approach methods, non-animal methods, neurotoxicity assessment model, next generation assessment methods, or nanotoxicity assessment method?
There can also be more than one single answer.
I guess we go to show the correct answers. And as Aisha mentioned, there are it can be new approach methods, non-animal methods, and next generation assessment methods. We go to the next question. What does process safety aim to prevent? Different possibilities for answer: fire, explosion, accidental release of substances and energies, or running out of coffee during safety meetings.
The process safety mainly aims to prevent also fire, explosion, accidental releases of substances and energies. So 3 of the options are correct. First 3.
Perfect. And we continue to this question, right?
Yes.
Yes. What does the abbreviation HAZOP mean? 4 possible answers: hazard and operability study, hazard analysis of zero operating pressure, hazardous operation and plant oversight program, or heat and zone optimization procedure. Okay, I will show the correct answer. And yes, it is hazard and operability study. And if I don't mistake, I give you the word back, right, Isa?
Now we are continuing with process-related safety. This diagram here is also a screenshot from SSbD framework, where we start with like understanding of the chemical and materials properties early on is essential, and then we are designing the processes and products. And in general, the starting point is the identification of the chemical hazards, which are then translated into process design and operations. And this means that the intrinsic properties, such as reactivity, stability, which we can assess using the in-chemical methods, directly inform the how processes are designed and managed. And a key tool here is, in this context, is HAZOP study. And later we will have an interactive session about it. And this is used to systematically identify the potential deviations in the process. This can be like changes in the flow, changes in the temperature or pressure, and it helps us to analyze the cause, the consequences of these deviations, and then we are reporting it to apply a safeguarding measure and actions. And the outcome, these assessments, feeds into occupational safety and health, environmental safety, and also other hazards. And then I will continue with the main pillars of the process safety. First, we are starting with the identifying the hazards. This means that we are analyzing the potential risks through the entire life cycle, but we are starting with the early design phase. And then once we continue with the startup and the operations, again, we can change it. And the second pillar is eliminate or reduce the hazards, ideally as early as possible. And this follows the concept of inherent safety and thinking early. For example, you can start with substitution, means that the hazards can be reduced if you can use a less hazardous material. This doesn't mean that you have to change your reactants. This can also be related to changing a cleaning agent for your reactants, for your reactor, for example, or for example, intensification. This can mean that storing your chemicals in a smaller storage tanks, for example, so that you can reduce the potential of hazards. For example, then attenuation means like operating under less extreme conditions. This can be like lower temperatures or pressures. And in addition, also hazards can be eliminated, they're limited by the containment or the separation. And the processes can be simplified, or you can try to avoid the human error there. And obviously, it is normal to not be able to eliminate all hazards from the system. That's why you should manage the remaining hazards. And this involves assessing the residual risk. Here we have another risk definition, likelihood multiplied by severity. And then in the end, we are documenting our assessments and communicating with our team. And another model that this is well known is called the SSbD model. There, the idea is to define that safety is ensured through multiple layers of protection, shown here with the slices, and the holes here represents the weaknesses. Like as long as the holes do not align, the hazards stop, and then they don't lead to any accidents. And however, if one layer fails, for example, and if all layers fail at the same time, an incident can occur. And these layers of protection can be grouped into 4: engineering controls, administrative controls, and behavioral controls, as well as the post-incident mitigation. Then now maybe we can continue with another mentee. Here, I can say that we will here now conduct a new HAZOP study together. And in the middle, we have our R101, the bromination reactant. And here we will think what could go wrong in this operation. We will only consider the reactor. We will only focus on one single node, because in HAZOP, we are studying one single node and assessing it. In another subject of HAZOP, we could also select one pipe, another valve, but this is only related to the reactor now. And then the next question can be like, what can be the cause? Or like, what can be the consequences of this malfunction? And how we can avoid it. These are the 4 questions that we should think of when we are going through a HAZOP study. So we talked about what could go wrong in this operation. Then the next question is what can be the cause? The third question, what can be the consequences? And the last question is how to avoid it. When we answer all these questions, we are essentially completing a mini HAZOP study. And to conclude of what HAZOP, how to conclude the HAZOP study, we should also look at the risk matrix, and this risk matrix is also available in the SSbD framework. And the next step is to determine how serious is this risk. And here is another risk definition, probability multiplied by severity. And in HAZOP, you can define your own ranking or your own likelihood. For example, I can say 5 per month. I can rank it as a 1, but some other person or for some other process, you can define 1 as once per 10 years. But in the end, you should report it. And we are talking about how likely it is, how bad it can be, and then we are finding the risk number here. And the last part, once we do this assessments, we can also how we can apply this into practice. There are available tools They're all available digital tools, and I just wanted to provide a screenshot from the Park toolbox. There are various process simulation tools like DNVGL, Asmentec, DWSIM, Honeywell, and ChemCat, for example. By using these tools, you can build a digital version of your process. You can simulate your temperature or pressure or flows. And in the end, you can test your different operating conditions and see what could happen if your temperature rises up, or what could change if your composition change, and these kind of things. So these tools are helping you to predict the risks before you build or operate your system. And with this, I have concluded my part.
Thank you very much, Aisha. And thank you all for your contributions to this very intense and very informative and highly valuable training session on safety dimension. So we took the next step now after the basic information we had last time, and we look very carefully now into hazard assessment more here now on the regulatory aspects of the QSA. Thank you to the QSA lab team for showing us the OECD guideline on the QSA regulatory aspects. Then thanks to Neeraj from TNO giving us insight into exposure modeling, and this was done in a very educational manner in my view. He also showed us the Stoffen manager, and then also Ponima. This was very valuable to look into environmental exposure modeling. We saw also the USTOX model shortly at least, I would say, but it was highly valuable insight in my view. Next time we are going to be seeing a little bit of EasyTalk, so we are looking into some modeling as well in further sessions. Thanks to Philip from BASF for showing us PBTK modeling, very important for extrapolating from in vitro results to in vivo concentrations and relating in vitro departure points to exposure doses that can be measured. Yeah, and exposure doses being measured. We were seeing this content presented by Mario from my team. And thank Aisha from BASF as well for giving us insight in quite a number of nice in chemical methodologies and even into process-related safety assessment. What's next? So we have done several sessions already. Please, if you feel also interested in getting more information on QSA, look it up in our resources on the homepage of Planets. This we will probably have to take on board for us in the project, and we will probably still come back to many of these content items that were presented today when we also discuss after the summer break tier 3 safety assessment methodologies, diving more deeply and more detailed into hazard and exposure assessments determination. With this, I would like to thank all the team that has been contributing today. We remain available for questions, comments, and concerns. info@planets.eu is our access point. And thanks to everybody. We are active here. Some networking avenues are listed here. Thanks everybody for today, and see you next time. Looking forward to the next events.